So today we have Daniele Diceo. He was in Malaga, no? And he's in Malaga, and then he made a short-term scientific mission with Joanna in Lisbon. And he will talk about advancing in vitro models of drug-induced liver injury, key techniques for the study of the immunological features of idiosyncratic hepatotoxicity in cell cultures. So if you want to tell us a little bit about your stay in Lisbon and how it helped you a little bit and how it made a change in your career, it would be excellent.
Of course.
But I think Joanna wanted to make a short introduction, no?
For sure.
Yes, I just would like to take the chance to just somehow tell you about our experience of hosting Danielle here in our lab. It was a really a great pleasure to have Danielle here, because there are only good things to say about him. He's a very gentle and intelligent person, really. So in the end, it made all of our work and life here very easy. So Danielle is very focused and very determined person. So he really knew what he wanted to do here with us. And he's not this kind of person that you just say, okay, we can make this experiment. If he doesn't understand or if he doesn't agree, he will not do it. So he really has to understand and to make sure that it makes sense and it's in accordance to his goals. So I'm really confident that Danielle will be a very successful researcher and he will have a very successful career. And I think for the short period of time that he was here with us, he managed to produce a lot of results that he will show us now. So I'm not going into details. I think it was a really, we really were able to change experience. Both of us profit with his training here. We profit with him. I think he profit with our experience as well. So I think it was very good, and it was again a pleasure to have you here and to receive you, Danielle. And you are welcome to come whenever you like, you know. So I think now I don't have much more to say, and I would like to hear what you have to say to us or to show to the rest of the people. Thank you so much, Danielle.
Thank you, Javier, and thank you, Joanna, for your words. For me, it's been a really enriching experience in the personal and the professional level. And what I'm going to present today is exactly that, my experience during a short-term scientific mission in your laboratory in Lisbon. But before in order to give a little bit of context to this experience, to the short-term scientific mission, first I want to explain very briefly what is DILI, although we already know, but also what I am doing in my PhD project. Well, most recent studies classify DILI into direct, idiosyncratic, or indirect. I don't want to go to many details again into what idiosyncratic DILI is, just to remind that it's an adverse liver reaction, consequence of the use of drugs or herbal products. And it has a low incidence in the general population. It is in general not dose dependent, unless we already know that a threshold dose is necessary for the injury to occur. It has the pathogenesis is believed to arise from the interaction of drug properties and host factors, such as the immune system, the microbiota, or the genetics. There is a lack of functional in vitro and in vivo models, which has a special impact during the preclinical development of drugs. And over 1,000 drugs are suspected to cause DILI, and is still one of the major causes of drug withdrawal from market and limitation of pharmacological indications during even clinical trials. It has a wide range of phenotypes and severity, being a frequent cause of acute liver failure. And finally, in the clinical practice, it's a complex disease because its diagnosis highly depends on casualty scores, and there is no diagnostic biomarker specific for this disease. As you may know, my PhD project is focused to the study of this DILI, and its provisional title is Immunological and Mechanistic Approaches to Drug-Induced Liver Injury, Determining the Immune Fingerprint of DILI using mass cytometry, in vitro characterization of mononuclear cell responses, and validation of an hepatotoxicity diagnostics test. As you may see, my PhD project is heavily focused to the study of the immune system in DILI. But why is the immune system so important? Well, as I said before, the pathogenesis of DILI is highly complex and is still not fully understood. The hepatocyte stress is supposed to be the first step in DILI. However, we already know that the stress produced by the drug or its reactive metabolites might not be sufficient to produce a clinically significant liver injury. There must be other factors that sensitize the hepatocytes to the injury in order for DILI to occur. And among these factors, both the innate immune response and adaptive immune response have gained increasing attention as a major driving forces leading to DILI onset and progression. In fact, different HL class 1 and 2 alleles have been identified as risk factors in the development of DILI, and there is growing evidence of for example, the role of the microbiota as its immunomodulatory capacities. Some authors specifically say that the injury followed by the drug-mediated injury of the hepatocyte is followed by the release of DAMs, these damage-associated molecular patterns that activate, for example, the Kupffer cells in the liver environment, which in turn release cytokines and chemokines to the liver, to the media. This induces a pro-inflammatory state and also recruits neutrophils and monocytes to the liver environment, further aggravating this innate immune response, which in turn amplify an inflammatory loop. These TAMs can also activate the dendritic cells, and the dendritic cells also produces co-stimulatory molecules, but very importantly, they are able to present drug-modified self-antigens. These activated dendritic cells might migrate to the lymph nodes and induce a drug-specific T-cell response. In the lymph nodes, the CD4 and CD8 T cells, recognizing these drug-modified self-antigens, can proliferate and differentiate. Once differentiated, they can migrate back to the blood, and in the liver, they exert different immunomodulatory functions. The effector CD4 T cells orchestrate the immune response, but are the CD4, CD8 T cells, which are the mostly cytotoxic, which kills the hepatocytes, producing a significant liver injury. Under normal circumstances, the cytokines and the cells involved in the immune tolerance might exert immunosuppressive functions, avoiding this to happen. But in DILI, this balance must be broken somehow, and it will be the result of an activated immune response and also a reduction of the regeneration, immune tolerance, or cytoprotection of the liver. Therefore, in order to deepen a little bit more in the immune mechanism of DILI, two of the main objectives of my PhD are focused towards the analysis of immune cell subpopulation in patients through mass cytometry, and also to perform a comparative in vitro analysis of lymphocyte activation by mitogens and LPS. This slide summarizes very well all my PhD project. In our group in Malaga, we recruit patients and also healthy controls and collect different biological samples, of which for this objective, the blood is the most important. Because from the blood, we perform the immunophenotyping of more than 40 different cell subsets of patients. And also we are able to do the lymphocyte activation assays through flow cytometry and Luminex technology. We believe that achieving these objectives will give us an unprecedented insight on the peripheral immune system in DILI. However, we also know that it's a huge lack on in vitro models of the immune system in DILI, especially in the preclinical setting. And also we know that further insights into the specific environment of the liver are needed. That's why we decided to add a fourth objective to my PhD named In Vitro DILI Models of Hepatocytes and Immune Cells. Our initial hypothesis was very simple. It was like in vitro treatment of hepatocytes with low doses of a given DILI drug can lead to the release of factors that trigger the activation of the immune system. In order to test this hypothesis, I contact Professor Joanna Miranda's lab, who leads the group of advanced cell models for predictive toxicology and cell-based therapies at the Faculty of Pharmacy of the University of Lisbon. And thanks to the cost priority DILI network, I was able to spend 4 wonderful months in her laboratory following a carefully designed work plan. Therefore, our general objective became to test the effect of conditioned media of treated hepatocytes on innate or adaptive immune cells. This slide summarizes very well the bullet points that I consider to be essential for understand all the methodology that I'm going to present later on. For all experiments, we used 3 different doses of one drug frequently associated with DILI, which is diclofenac. We use the therapeutic dose defined as the maximum non-toxic dose in cell cultures. We use the IC50, which is the dose that kills half of the cells. And we use water as control, as it is the drug vehicle. We tested all these different drug concentrations on 2 hepatic cell cultures, the G2 as a typically mortal hepatic cell line used in most toxicology studies, and the HLCs, which are human neonatal mesenchymal stem cells, reprogrammed and differentiated into hepatic-like cells. We tried to see an activation of the immune system using 2 different immune cell cultures, the PBMCs from human donors, and also the THP1 cell line, which is a human monocytic cell line derived from an acute monocytic leukemia patient. Therefore, our sub-objectives are to produce FG2 and HLC conditioned media using 3 different doses of diclofenac, analyze the effect of this drug directly on the hepatocytes, concentrate the conditioned media produced from these hepatocytes, and finally measure activation on the immune cells treated with either the drug directly, the conditioned media produced by the hepatocytes, or plasma from human patients. So the first thing we did was to calculate the therapeutic dose and the IC50 for diclofenac on FG2. For doing so, we performed 7 different dose response curves through MTS assays, obtaining a therapeutic dose of 0.025 and an IC50 of 0.53. For the case of the HLCs, it was not necessary to perform any dose response curve, as the values for the therapeutic dose and the IC50 were already calculated and published in a recent paper in the group. Based on these concentrations and after several adjustments and different tests, we decided that the best chronogram for performing injury assays on the liver cells was the following. After a treatment of the hepatic cell lines with the different doses of diclofenac, we will incubate them for 24 hours with the drug, and we will collect and store the supernatant after this drug exposure and add fresh media during an additional 8 hours. After these 8 hours with fresh media without diclofenac, we will collect the cells and the supernatant, which will be our conditioned media that we are going to use in the following experiments. Why do we choose this chronogram injury? Well, We believe that in the first 24 hours, the cells are in contact with the drug. For the same time, we used to analyze the drug response curve. So the injury, at least for the IC50, is guaranteed. In the next 8 hours with fresh media without the drug, we believe that the cells will still be releasing signals and dams that are of interest for see if they are able to stimulate an immune response. So the final work plan after the injury will be to perform a protein quantification of the assays and analyze the gene expression and cytokines production of the hepatic cell lines to assess the mechanism of injury of diclofenac on the cells. After this, we will concentrate the conditioned media and perform dose response curves of the immune cells with diclofenac. Finally, we will treat the immune cells with different treatment with the objective of assessing activation of the immune system through gene expression and cytokine production. Now I'm going to present the results for the diclofenac FG2 and immune cells experiments. The first thing I want to do is to share some pictures of how the injury assay looked. In this first example, we are going to compare the control condition to the therapeutic dose condition. And as you can see, after 24 hours of the treatment, the cells in the therapeutic dose, we don't see any morphological change or any noticeable cell death in the pictures. Even after 8 hours with clean media, we start seeing that the cells continue to grow. In this second example, at time 0, we are going to compare the therapeutic dose condition with the IC50 condition of diclofenac on FG2. This time at 8 hours, we start seeing some morphological changes in the cells treated with the IC50 with diclofenac, which became more evident after 24 hours of exposure with the drug. The cells start dying and the shape got more rounder. Let's remind these cells, we want them to be stressed as they will be our positive control of injury. Finally, after 8 hours of incubation with clean media, we start seeing that in the IC50 condition, some cells recover their shape, but many of them continue to die during this time. Now, after performing all the injuries assays and collecting the conditioned media, it is important to concentrate this conditioned media. But every treatment has to be concentrated in a different way, because the control condition might have, this conditioned media might have bioactive molecules produced by far more cells than, for example, the IC50. So if we do not concentrate it differently, we might find misleading results. For example, we might find that the control is stimulating more the immune system than the IC50, which is our positive control. So in order to solve this, we quantify the proteins of each injury assay in an independent manner to concentrate the conditioned media so they all can match a theoretically equal number of cells. So that's what we did. We performed the Bradford quantification of each injury assay. And as expected, we found that the IC50 had much less protein concentration. So based on these calculations, we were able to concentrate through centrifugation dynamic on the different conditions. So to have the therapeutic dose 50 times concentrated, the IC50 114 times concentrated, and the control 45 times concentrated. Besides the analysis of the picture, And we wanted to see the effect of diclofenac on the treated hepatocytes, FG2, through gene expression. We performed qPCR of 7 different genes, the TNF-alpha as a marker of a pro-inflammatory marker, the UGT2B7 is a phase 2 metabolizing enzyme. The BAX is related to apoptosis. SOT1 is related to cytosolic oxidative stress. ATF6 is related to endoplasmic reticulum stress. RIPK3 is related to necroptosis. And finally, PGC alpha is associated with mitochondrial biogenesis. All the results are referred to the control condition. We observe that the errors are very high, very big, because we are using, this is just a preliminary study. We are using a N very limited N. And also it's important to say that the selected genes were limited by the availability at that moment at the laboratory facilities. So the panel might need some adjustments. For example, instead of SOT1, it will be more interesting to analyze the expression of SOT2, as it is the mitochondrial isoform, which is the one more related to the hepatotoxicity by diclofenac. However, we saw that most genes seem to be overexpressed with diclofenac at the IC50 treatment, except for SOT1 and PGC alpha. Well, the next step was to treat the immune cells in order to see if we can induce an immune response, an immune activation. But which question do we want to answer by treating the cell this way? Well, the first one for me will be to know if the drug by itself is capable of stimulating an immune response, or for the contrary, it is necessary the mediation of the hepatocytes for this activation to happen. In second place, is the conditioned media of the therapeutic dose enough to induce an immune response, which is more related to a situation of idiosyncratic DILI, for example. And finally, we wanted to know if it is the plasma from DILI patients capable of stimulating an immune response different from the plasma from other patients, or for example, from healthy controls. To answer all these questions, we collected peripheral blood from healthy donors and extracted the PBMCs through fecal, and exposed these PBMCs to different treatments. We treated some of the PBMCs directly with the diclofenac at the 3 different doses, the same we use for the hepatocytes. We also treated the PBMCs with the conditioned media we produced after injuring the FG2 with the 3 different doses of diclofenac. And finally, we treated these PBMCs with human plasma from patients, both DILI patients and also other acute liver injury not related to DILI patients and healthy controls. After 24 hours incubation with all these treatments, we collected the cells which will be used for gene expression analysis, which is still pending. But the supernatant, we wanted to use it to measure markers of activation of the immune system. On these preliminary experiments, we test 2 kits of ELISA for detecting TNF-alpha and interferon gamma in the supernatant of the immune cells. We found out that for the TNF-alpha, all samples were under the lower detection limit of the assay, except for the PBMCs treated with the conditioned media of FG2 treated with the IC50 of diclofenac. This result for me is a little bit interesting because we couldn't see any TNF-alpha in the samples of PBMCs treated with the IC50 of the diclofenac directly, indicating that the mediation of the hepatocytes might be needed for the secretion of these pro-inflammatory cytokines in the PBMCs. However, no sample had detectable absorbance above the lower limit of the assay or the interferon gamma. So we had to ask ourselves, is the technique not sensitive enough? Or it is the sample that we use, because we use healthy donors, maybe they are not supposed to be activated by this methodology. So if it is the problem of the methodology of the technique, we found a possible solution to this experiment. It would be to use a Luminex panel. We found this Legendplex human inflammation panel, which has 10 times more sensitivity than the ELISA's kit for the detection of the interferon gamma and TNF-alpha, for example. Another possible solution we are considering is to use the conditioning media that we recover directly after the 24 hours incubation with the drug. This media may probably contain more signals or source signals than the one after 8 hours of incubation with clean media, and possibly be able to stimulate more strongly the immune response. Now I'm going to present the results related to the diclofenac on HLCs. The HLC's differentiation process is very sensitive and it takes time. We start from healthy donors, umbilical cord cells, and after 27 days of an optimized differentiation protocol, we achieved fully functional hepatic cell-like cells. And these are some pictures we took on the different HLC's differentiation process from day 0 to day 27, starting from the stem cells to a morphology very close to the morphology expected from hepatocytes. And the use of this kind of cells has many advantages over the use of immortalized cell lines. They tend to preserve more the drug metabolizing functions, and therefore will be more similar to the in vivo condition. This is just an example of the injury assay we performed with the HLCs. This is how they looked at time 0. And at 8 hours, we are comparing the therapeutic dose of diclofenac and the IC50 of diclofenac. And we can see already at 8 hours, we see a lot of cell death and also a very clear morphology change in the IC50 condition. After 24 hours of exposure with the drug, we continue to see these morphology changes and the death of many more cells. After 8 hours with clean media, we see that some of the cells with the IC50 condition start to recover their shape, but many of them continue to die. So the last thing we did was to quantify the protein of this injury assay, but as the differentiation protocol of HLCs, it takes time. I couldn't have more time to finish this part of the experiment, and no assays has yet been performed with these cells of the conditioning media, despite we have some samples stored. Some final thoughts about the experiments we carry out would be to say that I think we have established a very improved and standardized methodology for testing hepatocyte supernatant on immune cells. I believe that the use of conditioning media from treated hepatocytes might be a useful tool for the preclinical detection of daily drugs. Very importantly, we have many proteins, cytokines, and gene expression analysis still pending, and we have a lot of samples stored from this stay in Lisbon that we are planning to analyze soon. And we are also planning to incorporate the PBMCs from DILI patients as another group, not only healthy PBMCs patients. We believe that the incorporation of monocytic cell lines and other drugs, such as non-hepatotoxic drugs, will be also an interesting approach. And of course, in the future, the inclusion of 3D cell configurations will be optimal, will be more optimal. Where are we now? Well, we already have all the samples. Joanna sent us to Malaga, but we are waiting for all the material to arrive in order to be able to continue all the analysis and the gene expression experiments with immune cells. Just before finish, I want to take this opportunity to say that I feel very fortunate and thankful to Professor Joanna and all her team, from Nuno, Sergio, Joanna, Anna, and everyone else, Rita, that were very supportive with me, taught me a lot about all this methodology, and for me is the most important part of this experience. And everything went well in a very friendly way, so I have a good memory of all of these months. So thank you for the rest for your attention. If you have any question or suggestions, please feel free to ask me.
Thank you, Daniel. This was a very successful short-term scientific mission, because in such a small amount of time, very short actually, you produce a lot of data. I think the data are really important because there are not so many publications on the role of DILI, and specifically on diclofenac and on the immune system, as you mentioned at the beginning, no? Especially with the adaptive and the innate immune response, no? Yes. So I wanted to ask you a few things about this. There are some papers discussing about from the TACE group about as you mentioned, no, CCL5, some chemokines and their role in APAP, no, DILI. But do you think that dendritic cells and Kupffer cells play a major role rather than hepatocytes in DILI?
I believe so, because there is for the bibliography, I understand that recently there is growing evidence on the role of the innate immune response, and for us will be very interesting to include the Kupffer cells as a cell line to use for this experiment, to test them with the conditioned media of hepatocytes, and of course they might play a major role. Yes.
Yeah, this is what you showed at the end, no? That you want to use THP1. These
yes.
are monocytes that you can use basically as macrophages, no?
Yes, exactly. We want to differentiate them into macrophages just to see how they react to this conditioned media or either the drug alone.
So you showed that what is the difference between APAP metabolism in HepG2 and diclofenac metabolism? So because you showed that at different time points, yes. What is the main difference regarding cytochrome P450 metabolism?
I am not completely sure about how these drugs are metabolized exactly. We know that the injury assays had to be performed differently because the cells didn't react the same way when we treated, for example, with the IC50 of the drugs. That we had to make several adjustments to the injury assays. For example, with APAP, the cells at 8 hours was enough to kill half of the cells, but for diclofenac it wasn't. So we had to adjust it to 24 hours in order to see really half of the cells dying with the IC50.
Yeah, but you nicely showed then at this time point that you have RIP kinase activation, no, and
yes.
Endoplasmic reticulum at this time point, which is very interesting.
Yes, we believe that the mechanism of cell death with diclofenac is related more to necroptosis rather to apoptosis, but I will have to check that.
Yeah, this is quite interesting. Okay. Yeah, so please, someone else can ask some more questions. Any other questions?
I mean, Javier, we also know that diclofenac is the phase 2 metabolism is very important in diclofenac, and so this is also one of the reasons we think we have this difference with the cells. It's different from APAP. So PAP is mostly phase 1, and diclofenac, the hepatotoxicity is also much associated with phase 2. So it might need more time. And also we see the cells, these HLCs, they are more competent in terms of metabolism, and that's why they suffer more than the FG2 cells.
Yeah, this is I mean, really surprising.
So it somehow give us confidence regarding the use of HLCs for this experiment. But
absolutely,
I mean, it's, as you said, it's only three months or 4 months, and still Danielle produced a lot of data that
yes, and a lot of samples we have that haven't been included into results because we haven't analyzed it yet, but we will in the coming months.
And
so hopefully we're gonna find more interesting results.
We have Anna here also doing some more incubations with the diclofenac, so hopefully we will manage to have a bigger N.
Is there any animal model with the diclofenac that you can use? So I mean, in presence of the normal immune system, just to check when you induce the damage,
yes,
and see what is the response of the activation of the Kupffer cells M1 M2 response.
Yes. I know for sure there are models of diclofenac, animal models. I was trying to look for the simplest one, just to having an idea of what to do next. And I saw, for example, models in zebrafish studying the immune system of this fish treated with diclofenac, just to be the simplest approach following the in vitro conditions. But we haven't worked yet with animals, so it will be something to look forward to, or to perform a collaboration. I think it will be great.
Yeah. This you can characterize a little bit better in the presence. I mean, of course, in culture you can also use the THP1 and like you put in the last slide, but in vivo you get a full response, no, from the whole
exactly
active immune system. Yeah, I think so. Any comments, Maribel? The microphone.
Okay.
Yes, I think that Danielle did very, very productive stay during in Portugal. Indeed, it was really amazing the amount of data produced. I think it's very important to approach and to see how the innate response is modified. So the methodology that has been developed is very interesting. We are really very much looking forward to the results that will come out of this analysis. And as Joanna has pointed out, in one of the former studies to try to find out the genetic factors involved in diclofenac DILI, I remember that it was a paper by Guru Aithal published in Gastroenterology, in which the phase 2 polymorphism uridyl glucuronil transferase was much more involved in drug-induced in diclofenac DILI that the involvement of cytochrome 2C9 or 2C19. And also in another study done by us, the polymorphism of those cytochromes did not play any role in DILI development. So the results are in the really speak out of these differences in metabolism. And also what they saw is the production of adults, drug adults. And I don't know if that's something
Yes.
that, yes.
We thought about that, about testing if diclofenac was producing adults, which is this modified self-antigens. There is a way to do so. We found a paper describing that using antibodies target to the drug, we could, by Western blotting, know if the proteins are attached to the drug after removing the treatment. However, the antibodies for diclofenac at least are not standardized, the use of them for molecular biology. So it will be a very complicated procedure to produce them by our own. It will be something interesting to test in other drugs, which have the availability of these antibodies, but for diclofenac is not commercially available.
I will take also the opportunity to thank Joanna publicly for her friendship and good spirit and influential support and methodology approach that she inspired to Daniel. He was really he is actually very much motivated and willing to finish all these data and to analyze and to have results that can be shared with all of you.
Exactly.
It was our pleasure.
I don't know if anyone from the audience would like to make
any comments?
Any comments? Oleg, do you think that with your methodology, the methodology that you use, we could see or no?
Yeah, very nice presentation. Maybe one comment. THP1 cells are nice, but replicating the macrophages from THP1 might be challenging. Maybe consider some monocytes isolated from blood. That would be more relevant to have the actual macrophages, because even PMA differentiation of THP1 doesn't guarantee you to have the real macrophages.
Okay.
Yeah, and of course, the microscopy methods are very welcome to help provide any support, whatever someone envisions.
Yeah, that would be very, very important, Daniel.
Yes, we didn't thought about isolating monocytes from blood, but we have the capacity to do so. We have the access to the human samples, so maybe it's something we can seriously consider to do.
Yeah, it can be relatively easy. You can contact me. We have established protocol for the differentiation of the macrophages from monocytes, and from monocytes you can also differentiate as well the dendritic cells as well.
Yes.
Yeah.
That would be great. Yes.
Thank you, Oleg.
Thank you.
I don't know if Marina wants to add.
Hi.
Hello.
Hi. No, only to say that when Daniel came back from Lisbon, he was so motivated, and he only said, I need to continue this work because it has been great. So we started to plan in January, February, all the possible experiments that we wanted to do. The main problem we are having, I think all of you are having now, is a problem with the transport and the stock of some materials that is impossible to get. I don't know if it's the war or COVID or I don't know what exactly, but this is the biggest problem we are having. Not the experiments in our heads, and it's only a matter of the stock of some material. But yes, we think that this methodology could be used with other drugs, not also diclofenac. And I was thinking about the antibody about diclofenac. It's not that it's not available; it's available. We found one. The problem is that it's not tested for Western blotting. We contacted to the company, and they said that they did not recommend it to use this antibody. So, as Daniel said, maybe there are other antibodies to other drugs to see this mechanism of the formation of adults, that it's validated, but in the case of diclofenac, it's not validated. Maybe we can see it, but it's so expensive, and we didn't know if just the way to complete that those experiments. So I'm a colleague of Daniel, and I'm only to say that I'm very proud of him. It's an excellent colleague, and I'm so excited to complete these experiments and to see what happens in the future.
Thank you, Marina, for your words.
You're welcome.
But these adducts, you can see them as well with with metabolomics, right?
I think so, but the problem is that in Malaga we don't have the facility. We can do it because we are doing metabolomics for general metabolomics to study DILI, but we didn't think about using metabolomics to see the adults. So it's a good idea to think about it.
Yes, you can send it to Juanma.
Oh, great! Yeah, I know.
There are also some maybe Chinese colleagues, Jiajun Li, Jianxi.
Yes, we have Xiaohong Lei, Xiaoyong Li. Sanzi, Yanzi.
Javier, I was thinking about what you just said about detecting the adducts with metabolomics, and I don't exactly understand how can you detect this drug and protein interaction with metabolomics.
I think I saw a paper. Maybe I can send it to you. I think we via high throughput metabolomics. Okay. You can check interactions between drugs and proteins. Okay. But you need high throughput. Yeah, the normal metabolomics, I think you cannot get them.
Okay, I will check on that.
Ismael, I think the group by Nashville, Team Nashville, in Liverpool, they were approaching us because they wanted to measure amoxiclavulanate adults.
Yeah.
So they might have also some knowledge about this, or even Guru Aifa.
Yeah, Guru, yeah, for sure. Yeah. Yeah, I think the work is really interesting. I'm not so sure that you can. I mean, I'm sure that of course the innate immune system is very important, but you can also. But as you mentioned at the beginning of the presentation, the adaptive immune system is also involved because the effector T cells are the ones giving the response in the hepatocyte.
Exactly.
Yes.
And we also don't know if the T memory cells or T helper cells, circulating cells, I mean, are also intervening in this reaction. So using the PBMCs, I still consider it
makes a lot of sense. Yeah.
And we don't have to forget that in PBMCs also the monocytes are included. So this innate response hopefully is not hidden in the PBMCs, but it is also present.
Yeah, I agree. I agree with that. Yeah, I mean, at the end, it would be really nice to see one of these single cell sequencing, no, RNA sequencing with when you give the diclofenac and you see which cells are responding, and then you can really dig into specific molecular mechanisms.
Exactly, that would be amazing.
Yes, maybe you could comment, Daniel, why did you choose the diclofenac?
Well, we chose diclofenac basically because we knew that is one of the most frequent drugs associated with DILI, but also there is some description in bibliography that it might have some immuno-related effects on patients, especially related to dress, skin reaction, and other reactions. So we believe that if we had to choose just one drug for the stay, this could have more probability to find something related to the immune system. We tried also to use carbamazepine, but it is really hard to dissolve this drug on any solvents without being toxic to cells. So it is also another option we want to explore in the future to find a way to really get to dissolve these very apolar drugs.
But this diclofenac, as Maribel mentioned, is metabolized by this uridine gluconosyl transferase, right? So yes. This is like bilirubin metabolism.
Mm-hmm.
So does it trigger icteris? No,
but it's mainly in hepatocellular damage.
It's mainly
hepatocellular damage. Very rarely you get cholestatic damage. It's mainly hepatocellular.
Okay.